Hello, my name is Ron Shook, and today we are going to amplify the VRS1 gene in the Barley genome. There are two alleles of equal size base pair lengths. 2 rows is the dominant allele, and 6 row would be the trait expressed by the recessive allele. And I'm going to mix up some primers first. We'll just show you that I've set up my workstation. I have PCR tubes with tack beads already. I have my forward and reverse primers labeled and ready on ice. I will be making a mixture with this molecular water. I have a container full of my student products. They're Barley DNA, the template DNA right there. I have a number of pipetters set out, and I've already set them to the amounts I need. And along with those pipetters, I have the appropriate pipette tops. I can get all of my primer mixture into one 1.5 milliliter microtube, and I will start that off with 621 microliters of molecular water. So I'll get a tip on here. I told you that I had previously got this properly set to 621 microliters. We'll withdraw the liquid into here, cap my water so I don't spill, and express the water into the microtube. Alright, so we're good on the water. Dispose of that pipette tip. We need to add some forward and reverse primers. We'll be using a 2 and 20 microliter pipette for that. I measured out 10 microliters of forward and reverse primers back in the lab. So let's go here. This is my forward. I try to stay in the habit of always doing forward primers first. And we're set at 10 microliters here. And sure enough, we are going to get that. Always a good idea to watch the primer squeeze out. So here we go. I'll get that in there, and there is some of the forward primer. We'll, as long as we've got this pipetter, get an equal amount of the reverse primer in there. Let's make sure I got a nice clean tip. So 10 microliters coming up here of reverse primer. And again, we will make sure that we get that put properly into our water mix. And yes, indeed, we got that out. We'll come back in just a minute after I've finished off my pipetting. So I've completed my pipetting. Now I'm going to make sure my water and primers are well mixed. So we do a little vortexing. Okay, so we're well mixed, and I can just tap that down. Sometimes you can do a quick spin if you want, but tapping things down I think is going to do just fine in this case. So we'll begin getting some samples ready for PCR. I'm going to go through this next part methodically. The method will be that I will take my primer mix and each of my PCR tubes, which you can see here with the little white tack beads in them already. I'll pipette 24 microliters of the primer mix into each PCR tube. That will initiate some chemical reactions. I will then add 1 microliter of the template DNA from a particular class section, changing tips between each variety of barley that we are trying to amplify the VRS1 gene for and those associated alleles. And we'll end up with a total of 25 microliters per PCR tube. And then I'll put all the PCR tubes on ice or back in the fridge until I get another section done. But I don't want too much chemistry happening before I get the template DNA along with the primers into the PCR machine. So I've just finished pipetting the template DNA from our barley, 1 microliter of that, with 24 microliters of primer mix on top of a tack bead. I vortexed the PCR tubes that had all of those things in it. I made sure the tops of my PCR tubes were well positioned and that they were on tight. And I tapped them down so the reaction mixture will be at the bottom of the PCR tubes. There we go. And I like to do things like always have the low numbers to the left and closest to me. So my section 2 is at the bottom, section 8 at the top. Table 1s are at the left, table 7s at the right. And yes, my 8th section is missing 1 table. But now this is ready for us to close the top. Again, I've checked the PCR tubes to make sure the tops are fastened. We'll be bringing down this thermocycler top here. And when I push, that's my first close. Then I tighten about a quarter turn. I come down here. I am presently viewing the protocol for the VRS1 gene. What I will do is get this set and run the protocol.